Now we will discuss about non-cyclic electron transport system. Non-cyclic electron transport system, also known as Z scheme and photophosphorization. In non-cyclic electron transport system, both pigment system 1 and pigment system 2 involved. Photo system 2 means P680 consists of chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D and carotenoids. Phycobilins are also present in pigment system 2. As we know, phycobilins present only in pigment system 2. Phycobilins are absent in pigment system 1. Non-cyclic electron transport system occurs only in granum of thylakoids. Electron released by pigment system 1, electron released by pigment system 2 never back to it, and finally gained by NADP. NADP, N-nicotinamide adenine dinucleotide phosphate. Thus, gap of electron in pigment system 2 is filled by photolysis of water. And hence, after this reaction, oxygen is evolved. During non-cyclic electron transport system, approximately 3 ATP and 2 NADPH2 is produced. Four molecules of oxygen is photolyzed and one oxygen released. So here 12 NADPH2 and 18 ATP are required as assimilatory power. To produce one molecule of glucose. In dark reaction. Thus, 6 terms of Z scheme are necessary. If we see the structure of thylakoid membranes, this structure is known as lumen and this is thylakoid membrane. It is proteinaceous fluid known as stroma. In non-cyclical electron transport system, both pigment system 1 and pigment system 2 involved. Pigment system 2 means P680. When sunlight trapped on P680, electron released. And the first electron acceptor in non-cyclical electron transport system is thiophytin. So thiophytin receive the electron released from P680. Pheophytin transfer these electrons to plastoquinone. Plastoquinone pass to cytochrome B and then cytochrome F. Cytochrome F in the last transfer the electron to plastocyanine. Plastocyanine transfer the electron to pigment system 1, means P700. Here these electrons accepted by FRS. FRS transfer these electrons to ferredoxin Ft and Ft transfer to these electrons to stroma portion. In the stroma, NADP in the presence of protons converted into NADPH. NADPH plus HN in the presence of enzyme NADP reductase. NADP reductase. Due to the transfer of electron from P680, pigment system 2 is unstable. This stability is done by photolysis of water. In the lumen portion, 4H2O converted into electron, oxygen and protons, and this reaction is known as photolysis of water. These electrons transfer to pigment system 2 and hence pigment system 2 is stable. Plastoquinone helps in the transportation of proton from stroma to the lumen. So in the lumen portion there are accumulation of protons. Due to the accumulation of proton, there is a proton motive force occur. Proton motive force occur. These protons transfer by the coupling factor D protons CF0, CF1 means coupling factor 0 and coupling factor 1. Here ATP synthase is present. In the presence of ATP synthase, ADP plus inorganic phosphate converted into active ATP, and so in non-cyclical process ATP and NADPH asymmetry power are. So the asymmetry power is known as ATP and NADPH2. Pheophytin is a structure like chlorophyll A without magnesium. Pheophytin is first electron acceptor in non-cyclic electron transport system. NADP is known as final electron acceptor in non-cyclic electron transport system. And in non-cyclic electron transport system, energy flow from pigment system 2 to pigment system 1. Now we will discuss about dark reaction. Dark reaction is also known as thermochemical reaction. Dark reaction was first of all discovered by Melvin, Calvin and Benson. The site of dark reaction is known as stroma. In the dark reaction, at first RUDP combined with carbon dioxide and water in the presence of enzyme Rubisco converted into 3-PGA, phosphoglyceric acid. Phosphoglyceric acid converted into phosphoglyceraldehyde in the presence of NADPH2 and ATP. Phosphoglyceraldehyde regenerate RUBP and help in the formation of glucose. These glucose converted into starch. These are the food material for plants. This cycle is known as Calvin cycle. In the Calvin cycle, 18 ATP and 12 NADPH2 are consumed for fixation of 6 molecules of carbon dioxide. Dark reaction is also known as Blackman reaction or C3 cycle. In detail, the dark reaction, then we can say RUBP means ribulose biphosphate converted into 3-phosphoglyceric acid. 3-phosphoglyceric acid in the presence of enzyme Rubisco and carbon dioxide. 3-phosphoglyceric acid converted into 1,3-biphosphoglyceric acid. In the presence of enzyme transkinase, here 12 ATP converted into 12 ADP plus inorganic phosphate. 1,3-biphosphoglyceric acid converted into 3-phosphoglyceraldehyde. In the presence of enzyme dehydrogenase, here 12 NADPH2 utilized. Three phosphoglyceraldehyde converted into fructose 6-phosphate. Fructose 6-phosphate converted into glucose. And 3-phosphoglyceraldehyde converted into fructose 6-phosphate in the presence of enzyme aldolase. 3-phosphoglyceraldehyde regenerates glucose 5-phosphate in the presence of enzyme transketolase. Xylulose 5-phosphate converted into ribulose 5-phosphate in the presence of enzyme epimerase. Ribulose 5-phosphate converted into ribulose biphosphate in the presence of enzyme kinase. Here 6 ATP consumed. There are some factors which are responsible for affecting the photosynthesis. Means factors affecting the photosynthesis. First factor is light. Plants utilize sunlight. Also performing artificial light. Marine algae perform photosynthesis in moonlight. Very high light intensity cause reduction in rate of photosynthesis. Due to the denaturation of chlorophyll molecules. The second factor is light. The second factor is temperature. Optimum temperature range for photosynthesis is 25 to 30 degrees centigrade. Next factor is carbon dioxide. Carbon dioxide is known as limiting factor. It is first limiting factor for photosynthesis. It is about 0.03 to 0.1%. Next is oxygen. Higher concentration of oxygen reduce the rate of photosynthesis. Next is chlorophyll. Chlorophyll is essential factor for photosynthesis. Rate of photosynthesis is directly proportional to chlorophyll content. Now we discuss about significance of photosynthesis. It is essential for production of food material, photosynthesis also help in the atmospheric control and purification of air.